So now we will start our class. Okay, so we can go to the epigenetics. Okay, we'll go over. Okay, so for example, announcement of midterm exam. Okay, so I will explain the midterm exam once again. Okay, so midterm exam will be held on October 22nd, Friday, from 10:30 to unlimited. So usually maybe all of students maybe will solve the midterm exam within 1 hour. I think that I also think that you will also can solve all of midterm exam within 1 hour. However, our midterm exam time is unlimited. Okay, so take your time. Okay, unlimited. Okay. Okay. So test range is from the chapter 1 to chapter 5. Okay. So full score is 50. And also our homework is 50. So total, it will be the 100. Okay. So also this is test method. So we have to connect to the Joon. That this is important. You have to solve the test question while showing yourself in the real time through Zoom. So maybe everybody already finished another homework. So as a free check test for taking midterm exam number 2. So maybe all of the students already submitted their video. Okay, so they show that they are doing the computer, but they are recording their self. So it means that everybody is ready for the exam. Okay. Okay, and also another thing is that you can download. Okay, you can download the midterm exam, the file from the 10:30. So our exam will be start 10:30. So you have to enter Zoom class maybe 10:25 or 10:26, 10:27. Okay, not 10:30. Okay, because we will start our class 10:30. Okay. We will start our midterm exam from 10:30. So maybe you can attend the Zoom class around 10:25. So I will check your status. Okay, I will check that you are ready or you are not ready. You are ready or something like that. Okay. So and also, so you can print this file or write down the answer, or if you don't have your printer at your home, so you can write down the answer in the paper. That's all, okay? So, okay, and also this one is also important. After you finish midterm exam, so you have to write finished exam in the Zoom chatting windows. So we have the Zoom chatting window. So, okay, so for practice, you can write down finished exam right now. Okay. Okay. We have the chatting window. Now you can, for practice, you can write down finish it exam like this. Okay. Start it right now. Here, you can write down something like that. Please start it right now. Okay, like this. 천준영 finished exam. Okay, for example, T-board, you can write down T-board finished exam like this. Okay, so 김민주 finished exam like this. Okay, okay, 허재혁 finished exam. 김예슬미 finished exam is like this. Okay, so I will check it everything. Okay. Okay, like this, okay? So the important thing is you have to solve the test question while showing yourself in the real time through Zoom. And also, after you finish the exam, you have to write, for example, keyboard, finish the exam, or something like that, okay? In Zoom chatting window, okay? So I will check this, I will also check this one, okay? And after that, take a picture, answer sheet by your phone, send it to the Pervasive phone message or Pervasive KakaoTalk message within 5 minutes. So within 5 minutes. So I will also check this one. So I will check when you write down the finish the exam. So I will also check whether you submitted the answer sheet within 5 minutes, okay? So at that, make answer sheet as PDF version, as the same as the in homework. You can take a picture and convert it to PDF, or you can scan it and convert it to PDF. So finally, you have to submit answer sheet through assignment. Okay, any other question? So any other question about our exam, midterm exam? So everybody open the cam right now. 김유나, 김건영, 최윤기, 김혜선. 문경록, 이윤성, 김진웅. Okay, Zana, Marcin, Jana.
Yes.
Did you understand our midterm exam procedure?
Yeah, I understand.
Okay, you understand, okay. Okay, maybe everybody is understanding. Okay, 이윤성, 이수연, 문경록, 최세연, 김예슬미, open your cam. Okay. Tibor, did you understand all of midterm exam procedure?
Yeah, I understand.
Okay, you understand. Okay. Okay. And also question language for midterm exam. So questions in midterm exam are given in both Korean and English. Also, you can write down the answer in Korean or English. Okay, so don't worry. Okay. Okay, so this is also detailed information of midterm exam. So our total score is 100. Homework is from the chapter 1 to chapter 5. Total is the 50 points. So I checked that. Everybody already finished all of their homework. So everybody already got number 50. Okay. Okay. So today I found that Rayar and Marin and Ronika is not here. Okay. Okay, it's okay. Okay, and also this is detailed information of so from the chapter 1 to chapter 5. So we have the total problem number is 20 and total points is 50. Okay. Okay. So did you guys have any other question about the midterm exam? 김영석, 얼굴 보여?
네.
Did you understand all of the procedure?
네.
얼굴 보여? 이 이 매너가 이게 뭐야? 와, 정말 안 좋은 매너인데.
카메라가 밑에 있어가지고.
Okay. 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 Okay, so today we will cover maybe chapter 1 and chapter 2, maybe and part of chapter 3, okay? So now we will start chapter 1. Okay, so once again here. Okay, so our percentage of evaluation is that attendance is 20%, and midterm exam is 40%, and final term exam 40%. Okay, midterm exam is composed of the homework is 50, and midterm exam is 50, total 100. So 100 will be converted to 40% is like this, okay? So if you get the 95 or above, you will get A plus. I found that most of students got A plus. Okay. Now we will go to the chapter 1. So do you have any question in the chapter 1, in the homework or textbook, or do you have any question? So do you have any question in chapter 1? Okay, so now we will cover some important concept in chapter 1. So, okay, chapter 1, first one is so we learned about the centromere structure, and also especially we learned several centromere. For example, point centromere this is just a specialized DNA sequence. Its size is nearly the 125 base pair. So we can see the point centromere in the budding yeast, only in budding yeast. Okay. In chapter 2, we will cover the budding yeast. So usually in chapter, we will see the detailed information in the chapter 2, but I will cover here. So in the budding yeast, we will see the heterochromatin region in telomere region and around this centromere region. But actually, in the budding yeast, they have a point centromere. So actually, their centromere is not heterochromatin. But this region looks like the heterochromatin region because around this region, they have the MAT alpha or MATA, something like that. So they have because they have HML and HMR around the centromere region. That's why around the centromere region, it looks like the heterochromatin region. Okay, and euchromatin is located here and here. Okay. So, and also another concept is usually the most popular form of centromere is regional centromere. So this is regional centromere. However, the DNA sequence in the regional centromere is not conserved. Okay, and also we learned that we learned some concept of neo-centromere. So, and also DNA sequence in the neo-centromere is not conserved. And also synteny. So for example, synteny in centromere does not exist. So for example, centromere location in different So centromere location is different on the same chromosome within the phases. It means that synteny in centromere does not exist. Based on these three concepts, we can say that in chapter 1, we already solved the problem here. So we already provide 3 pieces of evidence that centromere is not determined by particular DNA sequence. So we cover there is no conservation DNA sequence in the original centromere and also neocentromere. There is no in neocentromere there is no similar DNA sequence, and also synteny in centromere. Synteny in centromere does not exist. Okay, and also epigenetics. We cover this definition of epigenetics, and also we solve this concept in homework 1. Okay, and also nowadays definition of epigenetics already changed. So this is modified epigenetics. So previously is the epigenetic landscape. So differentiated cells are unable to return to original pre-differentiated cell. They cannot go to the here. But nowadays, our definition of epigenetics is changes. So we call it as modified epigenetic landscape. For example, in the case of the pluripotent reprogramming, so differentiated cell can become the pre-differentiated, and finally they also become the pre-differentiated. They also can become the differentiated. And also there is another possibility. Direct conversion is like this: differentiated and differentiated, differentiated and differentiated, like this. Okay. So we cover here. We define the modern meaning of epigenetics, and also, so we cover the and also. Okay, also here, the important concept is the most important phenomenon in epigenetics is we can see in Argot-Rocros. So, okay, this is important, okay? So in midterm exam, I explained all of the background, okay? Important. So Argot-Rocros regulate production of the pure melanin. So pure melanin can make the yellow color, and the eumelanin can make the black colors. So it can determine the hair color in mice, especially the large allele A. So for example, large allele A can produce pure melanin. Most of them, they can produce pure melanin. So their coat color will be yellow. For example, small a, in case of small a is a mutant form of large allele A. So they cannot produce pure melanin. They only can produce eumelanin. So their color will be the black. So in the midterm exam, I explained this concept, okay? Okay, and also some transposable element was inserted into the promoter region of the allele, large allele A. So we call it as this one as the AVY genotype. So usually in the transposable element here, IAP, there are lots of CPG RNA is located there. So, for example, there is 2 cases when this is hypomethylated. So in these cases, it can activate. Hypomethylation still can activate the expression of large A allele. So it means that their coat colors will be the yellow is like this. But when their IAP is hypermethylated, when this region is hypermethylated, so it means that in these cases, the expression of the large allele A cannot be activated. So it can inactivate the expression of large A allele. So, but sometimes it can activate the large A allele during some specific point. That's why their coat color will be the brown is like this. Sometimes, but when the large A allele is frequently activated, their coat color is like this. But sometimes activated, their coat color will be brown like this. Okay, so you have to understand this concept to solve the midterm exam. Any other question? Okay, and also another example will be this one, the kin B. Okay, so this is also important. Okay, so this is fly larvae. They are same larvae, but when they eat the royal jelly, so this larvae will become the kin bee. When the same larvae eat the worker jelly, so they will become the worker bees like this. If you see some detailed information, so if they eat royal jelly, so in this case, this royal jelly can inactivate DNMT3. So for example, DNMT3 has 3A and 3B. Okay, so they can inactivate DNMT3. What's the main function of DNMT3? So this is the DNA methyltransferase. So in this case, so it can inactivate DNMT3. So it means that there will be no maturation. So it will become the kin bee. Okay. So when they eat a worker jelly, so it cannot. So when they eat the work belly, so it can it activates DNMT3. So it means that there will be lots of maturation. So it will become the work bees. So this is the epi allele, okay, a genetic locus which is affected by DNA maturation. Okay. Okay. So you have to explain you have to explain this phenomenon. Okay. Okay, we also cover DNA maturation. There is two kinds of DNA maturation. For example, DNMT1 is the recreate methylate pattern on only hemi-methylated DNAs. Okay. Another case is the DNMT3A and 3B. So their function is the de novo DNA maturation. So if there is no maturation here, but de novo maturation means that they can make each strand, they can make the maturation is like this. So we already stored this concept in the chapter 1. Okay. So describe the difference in the enzyme activity of DNMT1 and DNMT3 is like this. Okay. Okay. And also we learned about histone structure and also histone modification. Okay, so and also we cover histone code hypothesis. So we also cover this concept in the homework. For example, the concept of histone code hypothesis is that the transcription of genetic information is partly regulated by chemical modification in the histone tail. Okay, so for example, so they can be acetylated or they can be methylated. So there is some material that records the information of histone modification. For example, bromo domain. Bromo domain can decode the information. So this is the bromo domain can decode the information of the histone acetylation. So when another methylation, for example, histone methylation, for example, another chromo domain, total domain, MBT domain, another domain can decode information of histone methylation. So usually the histone nucleotide sequence is genetic information, but nucleosome can be the epigenetic information. So this is, we call it as histone code hypothesis. So we already solved this concept in chapter 1. So explain the histone code presented in 2001. So this is histone code hypothesis. Okay. Okay. Also, we also cover important, this is important, okay? Position effect variation, okay? Position effect variation. Important to solve the midterm exam, you have to understand this concept, okay? So usually, sometimes gene arrangement induces abnormal juxtaposition of gene with heterochromatin. So when the gene is put in the environment with a different condensation, so they will exhibit different phenotype. So this is position effect variation. Very important, you have to understand this concept. For example, in yeast, the ADE2 gene, so they can produce white cells like this. So they can make the white colony in yeast like this. So when this ADE gene is away from the telomere, so telomere is the heterochromatin region. Okay, so when they away from the telomere, this gene is away from telomere, so they cannot inhibit expression of the AD2 gene. So it will make the white colonies like this. So when when gene rearrangement is happens, so in this cases, ADE2 gene is next to the telomere, near to telomere. So in this case, it can inhibit the expression of ADE2 genes, so it will make the red. So red color with white sector like this. Okay. Okay. So this is the one example of the position effect variation. Okay. So there is another example of the eye color in Drosophila. So the concept is nearly similar to the yeast. So you have to know this concept. Okay, and also we also learn about some flexibility of heterochromatin formation. So this concept, with chapter 3, we learned detailed information of this concept. RNAi dependent heterochromatin formation. So usually during S phase, DNA synthesis is actively happen. During the DNA synthesis is happen, at that time mRNA synthesis is also actively happens. Okay. So in this cases, so based on this mechanism, so heterochromatin formation happens. So we will see the detailed information in chapter 3. Okay. And we also cover chip and also ChIP-seq, and also big data. Okay, we also learn the promoter structure of eukaryotic cells. Okay, and also NF1 region is the place where transcription factor can bind. Okay, so usually present in promoter region. So usually in this region, HAT, histone acetyltransferase, or another ATP-dependent chromatin remodeler can make this NF1 region. Okay, so in chapter 1 homework, we already cover in the multiple choices, we already cover this concept. Okay, so, okay, and also, okay, and also transcription initiation, because the RNA polymerase 2 they have the HAT function. Especially the RNA polymerase 2, they have a HAT function. For example, RNA polymerase 2, so they can induce histone acetylation here. So it means that they can make the nucleosome eviction. They can make the nucleosome eviction. So it means that after that they can initiate protein transcription initiation. So in chapter 1 homework, we cover this one. So the histone acetylation is very important during the eviction and the reassembly of the nucleosome by polymerase 2. Okay, because the RNA polymerase 2, they have the AHT function. So final concept, important thing is the pervasive transcription. So usually the pervasive transcription. So usually the only 2% genes that we are interested in are only 2% of genome. But transcription occurs in 93% of region in the genome. So it means that there is the pervasive transcription happens like this. So to prevent pervasive transcription, so we learned that if there is maturation, it happens in H3K36, so it means that they can recruit HDAC. Finally, it can prevent the pervasive transcription. So we also cover in chapter 1 homework. So for example, there is what aspect of gene expression when when there is a problem, the activity of the histone H3K36 methyltransferase, what happens? So maybe there will be the pervasive expression, pervasive transcription will happen. Like the answer is like this. Okay. Okay. So do you have any other question in chapter 1? Do you have any other question? Okay. So now we will go to the chapter 2. Okay. So, okay. Jana, Marcin Jana, do you have any other question in chapter 1?
No, that's good.
Okay, okay. Lonica.
No, I don't have any questions.
Okay, so maybe you missed the first part of our class. So I already recorded our Zoom class, so you can watch it later. Okay.
Okay, thank you very much.
And Marin and Raya is also okay.
Okay, thank you.
Okay, so in the first part, I explained some important concept. Okay, so Leia and the Martin and the Lonica, you both of them were late in the class, so you have to watch the video. Okay. Okay. So in chapter 2 is okay. So just a 1 minute break. So I want to drink some water. Okay, so 1 minute break. Okay. Okay. Okay, now I will start. Class. So Lonica, where are you right now? Are you in the Korea or are you still in the
In Korea.
Korea? Okay. So where do you stay, in the dormitory or?
No, we have an Airbnb apart and we got an issue with the Wi-Fi. That's why we couldn't connect.
Oh, okay. Okay. So Marine, so where are you right now? Are you seeing?
Same. I'm with Veronica.
Okay.
We are sharing a flat.
Okay. But in the exam, so you have to show yourself, okay?
Yeah, we
okay.
have separated rooms, so it won't be a problem.
Okay. Leia.
Yes, well, it's the same thing for me. I'm just in another room.
Okay. So actually, in the next Friday, we will have the actual exam. At that time, you have to show yourself, okay?
Yeah, okay.
Okay. 최세연, 수업 잘 듣고 있어?
네.
왜 숙제가 항상 늦지? 오케이? 주의하고 알겠지? 오케이. Okay, now we will go to the chapter 2. Do you have any other question in chapter 2? In homework or class, do you have any other question? Okay, now we will start chapter 2. Okay, so chapter 2 is the epigenetics in the budding yeast. Okay, so this is the life cycle or budding yeast, and also the important thing is the budding yeast, they have the point centromere. That's why they don't have so this region is centromere region, but actually the budding yeast, their centromere is not heterochromatin. Instead, around here, there are lots of the MAT locus or HML and HMR region. That's why this region looks like the heterochromatin region. So heterochromatin region is more condensed. Usually the gene silencing happens. Okay, the euchromatin is less condensed, and also gene is actively expressed. Okay, so and also we learned some the gene silencing mechanism in heterochromatin region. For example, heterochromatin formation is initiated at nucleation site. So they have some lipid, TG lipid, or some they have some silenced DNA, or they have some reparative DNAs, something like the TG lipid. For example, we already learned in the chapter 2. Okay, so in the heterochromatin formation, especially in telomere. So in the telomere region, they have a TG lipid. So at that time, in TG lipid, LAP1 and another protein is bind to the TG lipid region. Okay. Also, another concept is like this. So heterochromatin formation in HMR or HML. So in these cases, they have the E and I region. So usually in E and I region, so usually the ORC bind to E region, or ORC can bind to the I region. Okay, so they will make the they will make the heterochromatin formation happens. Finally, they will make the gene silencing. Okay, so general concept is like this. So usually the nucleation site happens, so they will make the nucleation formation. So some another enzyme and also silencing factor can bind to here. After they will make the spreading of heterochromatin. So nearby region will become the heterochromatin region. So in this case, transcriptional repression happens. So for example, some disease, for example, metabolic cardiomyopathy is due to heterochromatin due to the transcriptional repression happen due to some spreading of heterochromatin. So that's why some metabolic diseases happen is like this. So it means that some transcriptional repression can be inherited to your offspring. Do you have any other question? Okay. And also HML and HMR. So HML and HMR exist within heterochromatin region. So usually they are located around here. So we learned HML and HMR. So for example, HML have Y alpha gene and HMR has a YA gene. And also between the HML and HMR, there is a MAT. So this is a mating type determination locus. It's located between HML and HMR. Okay, so usually the HML and HMR, they are existed heterochromatin region. So they are the transcriptional inactive status like this. However, MAT locus is their transcriptional active status like this, and centromere is located here. Okay, and also so when the HML alpha is inserted to the MAT locus, so their mating type will be the alpha. So in this case, is MAT alpha 1 and alpha 2 can be expressed, or MARR is inserted to MAT, so they will MAT A1 and A2 can be expressed. Okay, and also regulation of mating type locus in the MAT A cell, and also we cover MAT alpha cell, and we cover MAT A and MAT alpha cell. Okay, how about in here? So in the homework, we cover this concept, okay? For example, MAT alpha cell, okay? For example, in MAT alpha cell, usually the MAT alpha 1 and MCM1 can bind to the promoter region of the alpha specific gene. So it means that the alpha specific gene can be expressed. However, in MAT alpha cell, they also have the A specific gene. But in this case, is MAT alpha 2 and the MCM1 bind to the promoter region. So they function as a repressor. So it means that A specific gene cannot be transcribed. Okay, so however, the haploid genes, so no binding to promoter. So usually the haploid specific gene can be expressed is like this. Okay, I will explain. Okay, I will explain some important concept. Okay, important. Okay, when there is mutation in the MCM1, it happens. So what happened to the alpha specific gene or A specific gene or haploid specific gene? What happened? Can you answer? 천준영, what happened?
네, 다시 한번 말씀해 주시겠어요? 제대로 못 들었어요. 다시.
Okay, very important. Okay, so I will explain it. Okay, so when the MCM1 is mutated, so it means that if there is mutation happen in the MCM1, so MCM1 did not function very well. Okay, so when mutation is happen in MCM1, so they cannot make the MAT alpha 2 and MCM2 complex. So it means that they cannot function as a repressor. However, MCM1, because there is a mutation is happened to MCM1, so they cannot also can bind to directly to the A specific gene. So it means that there will be no A specific gene transcription. So there will be no A specific gene transcription. The next case is that so usually the MAT alpha 1 and MCM1 they bind together. After that they function as activator. So when MCM1 mutation happens, so they cannot function as activator. So it means that alpha specific gene also cannot be transcribed. However, even though there is a MCM1 mutation is there, haploid specific gene can be actively transcribed. It is like this. Okay, I explained the very important phenomena. Okay, so actually our homework, I only give some question about it. So explain the expression of A specific gene and alpha specific gene and haploid specific gene in the MAT alpha cell. So the answer is like this. A specific gene is like this, and alpha specific gene is like this, and haploid specific gene is like this. But in MAT alpha cell, when the MCM1 mutation is happened, what happened to A specific gene, alpha specific gene, haploid specific gene? What happens? Okay, you have to know. Okay, very important. Okay, and also we learned the mating type changes in the wording is. So in this case, is the cell is that they are MAT A cell. So they in MAT locus they have A1, they have YA gene. So they can express the A1 and A2 together is like this. So, but usually the homologous recombination happens, the MATA can be changed to MAT alpha. Okay, so so before the making type changes, so their genotype is HML alpha, and the MATA and the HMA is like this. But making type changes frequently happened in the birding yeast. So at this case, RE, recombination enhancer, changes the chromatin structure. It means that allowing HMR alpha close to the HATA. At that time, transcription, homologous recombination happens between these regions. So finally, they will make the MAT alpha cells like this. So you have to understand this concept to solve the midterm exam, okay? Very important, okay? We already covered this concept in our homework, okay? So we already covered, described the mechanism of mating type change from MATA to MAT alpha in birding yeast. And it was very important, okay? And also we learned silent factor, and there is some difference between gene repression and the gene silencing. The most important thing is when SIR complex is there, this is recorded as gene silencing. When SIR complex is not present, we call it as gene repression. Okay. So this is SIR complex is like this one. Okay. So SIR complex. So usually the SIR 2, 3, 4, they make the 1:1:1 ratio. Okay. Especially the SIR2, they are function as the NAD dependent histone deacetylase. So they function as histone deacetylase. Okay. And also we cover this concept in chapter 2, multiple choice. Okay, here. So we call it in SIR complex, we call it as this one is trimer. So usually the SIR 2, 4, and 3 is like this, okay? Okay, and also we cover Heterochromatin Formation in telomere. Okay, and also we cover screening system to identify mutant causing gene silencing. Okay. First, we knock out URA3 gene in birding yeast. After that, we insert URA3 reporter system near the heterochromatin region. So they have the URA3, they have a specific function. It's like this. Okay. Okay, and also this is the one example of a simple reporter system using metabolic marker URA3. So if this is very important concept. So in midterm exam, so I explained this concept in the text. Okay, so also in homework, we already solved two problems regarding this question. This one, number 14 and number 15. Okay, very important, okay? So if transcription of URA3 reporter system is inhibited by something, it means that there is no URA3 expression. It means that no uracil synthesis. It means that no growth in the medium without uracil, but they have the resistance to 5-FOA. FOA means that fluorouric acid. Okay, the main function or another function of URA3 is they can change this 5OA to toxic product. Okay, because there is no URA3, so 5OA cannot be changed to toxic product. So they will have the resistance to 5OA. Another thing is that if transcription of a URA3 reporter system is not inhibited by something, so it means that URA3 expression happens. It means that uracil can be synthesized. It means that uracil, the URA3 can change it. Another function of URA3 can change it 5OA to toxic product. It means that they have no resistance to 5OA. Okay, so we already cover. So we already cover this concept. This is one case. This is another case. Okay, so for example, even though there is no uracil, they grow. The colony can be grow. If there is uracil, but also without the 5OA, they can grow. Okay, so if mutant grow in medium without uracil, but in this case, after we add 5OA, so they show that they did not show no resistance to 5OA. So in this case, this deleted gene is important for heterochromatin formation. Another case is this one. If mutant do not grow in the medium without uracil, but show resistance to 5OA, it means that deleted gene is not important for heterochromatin formation. Okay, so based on these two cases, you have to know. Okay, you have to know the function of deleted gene. Okay, any other question? This is very important. Simple reporter system using metabolic marker in the building yeast, especially the URA3. Any other question? Okay, and also we learned about another concept, simple reporter system using the metabolic marker HISPO. Okay. Okay, so this is the HISPO is located near the heterochromatin region. So we just inserted lateral transposon in the promote region of HISPO. Okay, so there will be two cases. Is like this. Okay, so we already solved this concept in here. We already in these cases we generate if mutant can grow in medium without histidine. What is the main function of deleted gene? So what is the main function of deleted gene? It's like this. So in these cases, so if mutant can grow in medium without histidine, it means that deleted gene is not deleted gene is important for chromatin remodeling. Okay, so I will explain this one. Okay. First, we can generate mutant in which certain gene is deleted. So if deleted gene is important for chromatin remodeling, means that there is no chromatin remodeling it happens. So in this case, if there is no chromatin remodeling, so chromatin remodeling means that from heterochromatin to euchromatin. So in this case is when this Ty is located within the heterochromatin region. So in this case is transcription within Ty is lost. However, the expression of the original His4 is revived. So His4 gene is actually located in the near the heterochromatin region. So they design, their function is sometimes they can be expressed in the heterochromatin region. Sometimes they cannot expressed in heterochromatin region. Okay, but sometimes they can be expressed. So it means that they can express His4. So they can grow in the medium without histidine. So if mutant can grow in the medium without histidine, it means that deleted gene is important for chromatin remodeling. This is the opposite case. So if mutants cannot grow in the medium without histidine, it means that the deleted gene is not important for chromatin remodeling. Very important. So you have to understand this concept to solve the midterm exam. Any other question? Very important, okay? And also we cover GCM5 and 3 and SNF complex. So this three gene is discovered using the simple reporter system using metabolic markers, especially His4. So we find the GCM5 and the function of we find the GCM5 and 3 and SNF gene like this. Okay. Okay, and also chapter 2 homework number 16 is very important, and the number 15 and 14 is also very important. So we cover heterochromatin formation, telomere, and also we cover the difference between the gene repression and gene silencing. And also mating type changes, very important. So you have to explain the procedure, okay? And also this is some species cases MAT alpha cell. So what happened when the MCM1 is mutated? What happened in MAT alpha cell? That one is very important, okay? And also we covered some important in multiple choices. We covered several important things, okay? Okay, any other question in chapter 2? The layer, did you understand all of concept in chapter 2? Or this concept in the concept in chapter 2, most of them is very difficult to understand, but you have to understand, okay, layer?
Oh, sorry, can you hear me?
Yeah, I can hear you.
Okay.
And also, okay, so because the concept in the epigenetics is very difficult, usually very difficult to understand. So that's why usually the epigenetics is not open in the undergraduate course, but I opened this course in undergraduate, okay? Okay. I try to explain it very simple, but some concept is very difficult to understand. You have to study, okay? Okay. Okay. So now is we have the 8 minutes left, okay? So now we can go to the chapter 3. Chapter 3 is the cover epigenetics in the fusion yeast. Do you have any question in the homework or the class? Do you have any question? The concept in chapter 3 is very difficult to understand. I know it's very difficult. So I try to explain it very simple. Okay. For example, birding yeast, they have the haploid is alpha, or they have the A cell. However, in the fission yeast, the haploid, they have the MAT2P, or they have the minus type is MAT3 minus 3M is like this. So usually the mating type region is also present in the heterochromatin region in fission yeast. Okay. Okay, and also their life cycle is composed of the interphase and M phase, and most of the cell cycle is G2 phase in the fission yeast. So this concept we already shown in the multiple choice. Okay, in multiple choice we learned that. Okay. Okay. No. Okay. 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 And also, okay, the important concept is, okay. So from fission yeast, heterochromatin region is located in the centromere. From fission yeast, centromere region is the heterochromatin region. And also the mating type locus is also belong to the heterochromatin region. So usually the in fission yeast, also the mating type locus is located near the centromere. In birding yeast is also mating type locus is located near the centromere region. Okay. Also genes involved in the meiosis is also located in heterochromatin region. Okay. Okay, so usually from fission yeast, the heterochromatin region is located in the centromere is heterochromatin region. For example, this is the regional centromere. Okay, so they have heterochromatin region is located here. Okay. And also from fission yeast, so they have the centromere structure like this. But this structure is the molecular mechanism is nearly similar to that seen in higher organism. For example, this is fission yeast and fruit flies and human, they are nearly similar to each other. Okay. So they have the central region and they also have out region. Centromere region, we call it as the kinetochore binding site, and also we call it as the stamp A binding region. So this is the stamp A nucleosome. This region is we call it as stamp A binding region. So they are located around here. And out region, we call it as heterochromatin region. They are composed of the more OLTL and OLTR. And each region is composed of DG or DH. Okay. So this is the important, okay. So this is very important concept in chapter 3. Okay, so this is experiment, very important. You have to understand this concept to solve the Maturation. Okay, so so this is OTR region. So this is the experiment to reveal the function of OTR sequence. So, okay, so this, for example, the OTR region is composed of DG repeat or DH region. Okay, especially in this case is the insert or part of OTR sequence, especially the fragment of DG. We call it as this one as L5 elements. So we insert this region to euchromatin region. So when this region is inserted into the euchromatin region, so usually in this region, they become the heterochromatin region. After that, it can initiate heterochromatin formation. Finally, it induces silencing. So based on this experiment, we can say that we don't know some specific mechanism, but in this experiment, we can say that maybe some special, some transcription factor. So we will see some detailed information in the later RNAi dependent heterochromatin formation. But based on this vision, this experiment, we can say that maybe some transcription factor can bind to the unique sequence in L5. So maybe this region, some transcription factor, they bind to the L5 element, maybe play an important role to form a heterochromatin region. Okay, so based on this experiment, we can say that like this. Okay, so detailed information we can see in the later slide. Okay, especially we call it as RNAi dependent heterochromatin formation. So do you have any other question? So okay, Lonica, Marine, and Raya. So you were late class. So I already recorded all of June class. So I will upload this class to our e-learning site, so you can watch it. Okay. So I explained some important concept in the first part. Okay, first part of our class. Okay. So do you have any other question? Any other question? Okay, so today we will finish class and this Friday. So we will cover part of chapter 3 and chapter 4 and chapter 5. Okay. Okay. Goodbye. Okay, so finish class. Goodbye.
감사합니다.
Okay.
감사합니다.
Thank you. Bye.
감사합니다.
Thank you.
Thank you. Bye.
Okay.